In this video, we will review the enzyme assignment extension questions. Number 20, thermophilic bacteria, such as Thermus aquaticus, live in hot springs where the temperature is greater than 70 degrees Celsius. Draw a graph similar to graph A in Model 2, representing the optimal temperature of this bacteria. So when we look at the rate of the enzyme activity, we need to have, if it lives in a hot spring where the temperature is greater than 70 degrees, that means that the starting point of our graph, where we see an increase, has to be at 70. So I'm going to say, so we've got 10, 20, 30, 40, 50, 60, 70, 80, 90, 100, we're going to say. So if I go to 70, that means our enzyme cannot start working until after 70. So our graph is going to be something like that, where this is 70 degrees. If the temperature in that hot spring is always greater than 70, that means our enzyme will not work below that. So that's what that graph would look like for the rate of the reaction. Number 21, DNA polymerase from the TAC bacteria is used in PCR, polymerase chain reaction. Polymerase chain reaction is a technique where millions of copies of DNA can be made from one original copy. In this method, the target DNA molecule is subjected to temperatures over 95 degrees Celsius to make the double-stranded DNA separate. The temperature is then lowered slightly to allow primers to anneal before the TAC polymerase catalyzes the reaction to incorporate new nucleotides into the complementary strands. The cycle is then repeated over and over until there are millions of copies of the target DNA. You have to predict why this bacterial polymerase is used instead of the human polymerase. So this process happens at temperatures over 95 degrees Celsius. So human body temperature is 37 degrees Celsius. So if we were to use a human enzyme, compared to 37 degrees Celsius, 95 is very, very high, and most likely our enzyme would be denatured as a result of that. So we're going to predict and say that the human polymerase denatures at 95 degrees Celsius. So if we were to use this enzyme, would we have a reaction? No, because the polymerase would have denatured. So you would have no replication of DNA due to the denatured enzyme. In these next questions, the rate of an enzyme-catalyzed reaction can be affected by a couple of different things. One of those is a coenzyme. A coenzyme, you want to think of it as something that will boost the enzyme's ability to work, and then an inhibitor is going to stop or prevent the enzyme from being able to work. So number 22, you have to sketch a graph that shows the relationship between the rate of an enzyme reaction and the concentration of a coenzyme that is necessary for the enzyme to function properly. So in our graph, if this is our coenzyme, and this is the rate of our reaction, we're going to have something that looks like this. The more coenzyme we add, the higher the rate of the reaction is, because the coenzyme is boosting the enzyme's ability to work. At some point, if we continue this graph on, we will have a leveling off because the enzyme will level off with its reaction. But a coenzyme will boost the activity, so it will increase the rate of reaction. Number 23, add a line to graph C of Model 2 that shows the rate of an enzyme reaction in the presence of inhibitor molecules. So our graph in Model 2 for C, for the substrate, for the concentration, looks something like this. If we add an inhibitor is going to stop or prevent the substrate from fitting into the active site of the enzyme. So if we were to look at that, we are going to have the inhibitor molecule is going to, at first we're going to have a little bit of reaction, and then it's going to drop down and completely stop as we add more inhibitor. So if this is the inhibitor, we're going to have a little reaction at first, and then once those inhibitors start filling in the active sites of our enzymes, it's going to stop our enzyme from being able to work anymore. And that is all of it. Hopefully this helped.